why this is all important. The investments in research, basic research, the investments in biotech and its contribution to society, and the whole training and supporting people. It's a very important story from that perspective.
Yeah, I think that's very important, especially given some of the misinformation about what RNA is and what RNA can do. And I think this talk is very useful because it shows how RNA has the potential to cure really difficult diseases and improve quality of life for everyone.
It doesn't have the potential. It has already demonstrated it's real.
Hello, and welcome to the International Business Today podcast. This is Professor Ravi Sarathy, Professor of Strategy and International Business at the Damore McKim School of Business at Northeastern University. My guest today is Professor Phillip Sharp, Institute Professor at MIT and co-founder of Alnylam, one of the first companies with an RNAi, RNA interference-based pharmaceutical drug approved by the FDA, and also a co-founder of Biogen, one of the very first drug companies in the biotech field in the world. He's also a Nobel Laureate. He was awarded the Nobel Prize in 1993 for some of his foundational work in understanding the structure of DNA and RNA. Phil, I'm delighted to have you here as our guest, and I would love to have our audience learn from you about yourself and your ability to straddle the worlds of science and industry. So welcome very much.
Well, it's a pleasure to be here.
Phil, it's not every day that we have a Nobel Prize winner at our podcast series. And it's a very rare event. There have been relatively few scientists, given the number of scientists all over the world who have won Nobel Prizes. So please tell me a little bit about what it takes to win a Nobel Prize, how it felt when you heard that you had won it, and what the ceremony was like when you actually attended the Nobel ceremonies in Sweden.
So the origin of the prize was early on in my transition to beginning to focus on the cell biology of human cells. I came to suspect that how genes were expressed through RNA to make protein was different in human or multicellular animals, organisms, than in bacteria. And it had been worked out in bacteria. So I began a research program using electron microscopy to look at RNA and DNA, and then discovered in 1977 that our genes come in pieces. They're not one contiguous sequence that is used to make the protein. That sequence comes in bits called exons, and you have to splice those bits together between the making of the RNA and its translation as a message. It was RNA splicing. Totally unsuspected. No one speculated on it. It was stunning. And that turned out to be terribly important in our understanding of how medicine works, how genes are expressed in proteins. It wasn't until 1993 that I got the Nobel Prize for that work with Rich Roberts at Cold Spring Harbor. He had done work, Cold Spring Harbor had done work in parallel. And the morning that the call came in, it was Columbus Day at that time in New England. It's called Columbus. And the call came in at 6 a.m. I pick up the telephone, and someone I knew from the Swedish Academy said, "You've been selected for a Nobel Prize." I was totally stunned. And the next question was, "Do you have the telephone number for Richard Roberts, the other co-winner with me?" And I couldn't even think. There was no way I was going to find his telephone number. So Rich and I received the prize. Sweden celebrates the Nobel Prizes as a national holiday. I mean, it's a national effort. You are hosted at the best museums and meals and events in individuals' homes, events at the palace with the king and queen at a large table. And the night after you get the prize, which is given in the opera house with a very formal celebration, you go to the city hall and 1,100 people have dinner with you, and with pageantry. It's just a very special experience. We were there for a week. I took my parents and some scientists from the laboratory that had done some of the work, and it was very special. Just so if they ever call you for a Nobel Prize, please don't turn them down.
Unlikely, but it's wonderful to hear about the experience, and I'm delighted that you had been able to do it. I think it's a well-deserved reward for all the amazing work you've done, not just in terms of pure science, but also the very critical fact that you've been able to help very much in translating that science into helping human lives. Now, one of the hardest things, I believe, in any science-based industry, but particularly biotech, is how to take science and make it into a business that is both commercially viable and very useful. Right? So can we talk about how did this idea of using RNAi to form a company happen?
Well, there was a discovery in 1998 by Andy Farr and Craig Mello that showed that double-strand RNA from a gene, which is not frequently found in nature, when introduced into a worm in this case, would silence the gene, specifically the gene in that worm tissue throughout the body. It was a totally unexpected discovery. And it raised the possibility that this mechanism or part of it, i.e. double-strand RNA would signal silencing a gene, could work in other tissues. And shortly thereafter, there was a demonstration that it worked in Drosophila, a fruit fly. So the idea that it might work in human cells was so exciting because we had no technology to silence a gene in mammalian cells in the laboratory or certainly in medicine. So with that discovery, the idea was if we understood the mechanism, then maybe we could then adapt this breakthrough to treating disease, silencing genes that cause disease.
Exactly. So what you're saying is many diseases are caused by certain genes, which are possibly mutations. And if we can somehow figure out which gene causes which disease and then use a small interfering RNA to silence that gene, then you've essentially solved that disease and come up with a treatment.
That was the exciting possibility. And so we set out to see if we identify the mechanism or elucidate the mechanism, which happened very rapidly in Drosophila cells, which again is not human. And then quickly, the group of us who was working together at that stage, one in the Max Planck in Germany, one in Cambridge, one at UMass, discovered the structure of the small RNA. And that really opened a direct channel to saying, if I can get this RNA into disease cells, I could probably change the course of disease. And that promise is what we wanted to translate to see if it would work in patients and benefit people.
Mm-hmm.
So just a little fast forward. Sure. I knew I needed to create a buzz, all right? So I contacted a company that was a very forward-looking company, Millennium, and asked them if I could come up and pitch this to Millennium. And I did. And they found a lot of it very exciting, but said this is too far down the road, too long before this could develop for us to be interested in. But it created a buzz in Cambridge that then allowed us to find venture capital and to begin to recruit people.
Mm-hmm. That's a very important point, recruiting people, because when you want to go from the lab to the world of industry, to the world of commerce, you need skills that are not only scientific skills. So how did you go about establishing this initial founding team where you need to balance science with corporate skills and corporate leadership? And you also need to think about costs. You have a budget. And so you need to think about how to do this economically and still achieve our goals.
Well, you have to depend on others for some of this. Mm-hmm. The first thing we needed to do was to identify a venture capitalist organization and principal who would share your vision of taking this technology all the way to a patient. And then, in this case, it was Polaris, who we selected as the first lead venture capitalist. There was Atlas as well involved and others. And Christoph Westphal, who I had known for a long time personally, was a principal there. And he, working with us, took a little incubator space, temporary space, and began to use a network to identify CEOs, candidate initial CEOs. And fortunately, from Millennium, John Maganori, who was there as a leader of Millennium, was interested in being the CEO of this new startup. And John joined us. And with some venture money, we were able to recruit a small team of 5 or 6 people. Most of the finance was contracted out for the first several years, and we had consultants for organizing and lawyers to form the paper. But the laboratory was a core of a few people who were assembling all the information, beginning to do a few experiments, looking at the intellectual property, negotiating the intellectual property with MIT and elsewhere, and beginning to organize the company. So you have very small amounts of resources that have to be used very effectively and focused on producing a company that had the freedom to operate, form relationships, and then recruit other people, but do it in the most prudent manner you could.
Mm-hmm. Now, you also have mentioned patents, the critical importance of patents when you're moving into a very new field, and you need to be able to control those patents. Could you briefly explain how you got the patents in place to be able to be sure that if you were successful, then Alnylam would be able to reap the benefits of its work?
Well, it's more important than just that. MIT filed patents on the initial science done in Drosophila, but that's not humans. And then Tom Tuschl, a fellow who was a co-founder of Alnylam, but at that stage was in Germany working with Max Planck, did a very direct experiment that showed that RNAi would work in mammalian cells. MIT and Max Planck jointly patented that material. And then there was other patents that were more theoretical than real, but they can complicate your life. And those we consolidated with the acquisition or relationship with Biopharm. And that gave us a very strong patent position. Now, this was a new science, RNAi, and pharmaceutical companies are very leery of committing resources in an area in which there is not patent position. So that patent position allowed us to form relationships with Novartis and Merck and others, and gave us the freedom to operate, meaning it was unlikely that someone would be able to step in and, for patent reasons, block the progress of the company. So the intellectual property is extraordinarily important in pharmaceuticals. It takes about 10 years of investment, in this case 16 years, before you get to market. And without the protection of a patent that says, when I get to market, I can be exclusive and actually reap some of that benefit in terms of the cost, is a very essential part of pharmaceuticals.
I believe related to this patent issue, you acquired the German company, Ribofarm.
Yep.
And you also licensed some technologies from Arbutus, what used to be Techmira, if I recall. Why was that so important at that early stage when you already had these strong patents?
Well, there was a patent we were concerned about in Ribofarm. It was part of the patents that we had as a portfolio. So we consolidated patents. We also got some expertise there. But in the latter case, Techmira and Arbutus, it was the technology that we needed to make delivery of these RNAs to patient cells work. And that's a very interesting development. Peter Cullis in Canada was one of the first to use a set of lipids of a certain nature to form a lipid nanoparticle. A little particle looks like a virus, same size as a virus, but you can have a payload inside that particle that you can deliver to cells. And that technology was very important in developing the siRNA delivery system we initially used to treat disease, because you had to get these small RNAs inside cells to work. And the cells do not take up RNA. So you had to package it like a virus and put it into cells.
Because if you didn't get the siRNA in the cell, it wouldn't be able to silence the gene.
It wouldn't silence the gene. The interesting thing, this was about 10 years before the COVID vaccine. And the same technology was the basis of the COVID vaccine. So that whole system of delivering RNA in the COVID vaccine, it was messenger RNA. In this case, it's siRNA. It's a whole different way of working, but the same particle technology was used.
Using the liquid nanoparticles.
Using the liquid nanoparticles. So science builds on itself in a very important way for society.
Now, clearly you were interested in treating diseases. How did you pick which diseases to target initially with this new idea of using silencing RNA?
Very important. The whole technology is gene-centric. And Alnylam was started in 2002, and the sequencing of the human genome was completed in 2003. So we had the sequence of the total human genome, all 23,000 genes, and therefore, if you can identify a disease related to the misactivity of a gene, then you can design a RNA based on the sequence of that gene to silence that gene. So here we needed disease, genes that cause the disease or was active in the disease, the sequence and design of the right RNA and delivery. That was the issue. Could we develop that system that would then allow us to treat disease?
So could I say that each gene is almost like it has its own special lock? And so you had to design a special silencing RNA that was the key for that particular gene and that could open the lock and then get inside the gene and silence it.
That's exactly right. But we do know now over 5,000 genes that cause disease when mutated in patients. That's from genetic diseases that are passed from father and mother to children. And we are now sequencing all sorts of tumors and other disease tissues. So the whole gene-centric approach to treating disease has exploded at the same time the RNAi technology became available, and that was all part of the momentum behind Alnylam.
So could you tell me a little bit about, say, a particular disease? You mentioned hybrid TTR as a disease that you targeted initially. Could you just briefly explain what that disease is and why that might work using the silencing RNA technique?
Well, there's a particular disease. It causes amyloid, which is misfolded protein, to accumulate in nerves and in heart. And in both cases, it will cause death of cells, your nerve cells in a neuropathy, and in cardiology, your muscle cells in the heart. And so that gene, when mutated of certain positions, misfolds in the blood, accumulates in those tissues, and causes the death of cells. So you lose the ability, if it's a cardiomyopathy, to walk upstairs. So we knew the gene, and the gene was produced, the protein from the gene was produced in the liver, and that's where this lipid nanoparticle technology was optimally active. So we knew the protein was produced in the liver, enters the blood, misfolds, accumulates, and destroys cells. So the issue was take the RNAi, deliver to the liver, and silence that gene. And that is the first drug that was approved for Alnylam in 16 years after the company was founded. They got FDA approval 16 years after.
Now, these are rare diseases. So we have in the U.S. something called the Orphan Drug Act, right? So how influential was the existence of the Orphan Drug Act in terms of Alnylam deciding how to proceed with drug development?
Well, it was useful because there's some advantages to being designated an orphan drug. 7 years of exclusivity, some resources that if it's sufficiently orphan, and then you can adapt. But this technology was so new, and the patents were so new, that we were primarily working on the intellectual property that we had developed over those 16 years to protect our therapeutic treatments and be able to sell products around the world. I mean, the patent structure in Europe is very similar. It's different. There's a different criteria. We're now closer to an international criteria than we used to be for patents. But you can get the same type of exclusivity for patents in the rest of the world as in the U.S. typically. And therefore, it becomes part of the ability to have a monopoly for a while to capture the resources needed to pay for the development of the technology.
So the 16 years of investment have to be recouped, and the 7-year exclusivity gives you a fighting chance.
Even 10 years or more for a patent.
Now, it's not a smooth path, right?
No.
So almost every drug company that is trying to do something pioneering faces reversals, runs out of money, or has the danger of running out of money before they get to the point where they can get a drug approved and start marketing it. So you have years of accumulating losses, negative cash flow, your cash runway diminishes. And revenue prospects are very far away. So how did Alnylam manage that process of not running out of cash even though perhaps, it's not a smooth path, 16 years clearly shows that it wasn't a smooth path.
Well, it wasn't a smooth path, and 16 years is a long time to support a company that it's not involved in sales. So early on, there was research agreements struck with Novartis, Merck, because those companies wanted to understand what RNAi meant in terms of both research as well as possible therapeutic endeavors. And that gave us, in those relationships, some support of ongoing research at Alnylam, as well as some upfront payment for the technology, the intellectual property, the value the company had. And then we signed agreements with other pharmaceutical companies with emerging data that said we're going to be able to, make advantages and make products in this space. Then we took some, debt. And we did an IPO and then a second IPO, offering of shares. We raised somewhere in excess of $2 billion.
$2 billion.
In excess. That's why it was so important to have an experienced management, a CEO, and a core of people who understood the technology. John Maganori is a PhD and a very successful scientist before he became a CEO, and a forward-looking finance, patent lawyer. All of it had to be together. It had to be a coherent, visionary leadership, as well as excellent science, committed to making this real, making RNAi a therapeutic to treat patients.
Now, since we're talking about this irregular path and a difficult path, obviously there were setbacks at various points where things didn't go the way you anticipated, and you found some new data, some new results that were, okay, we got to rethink how we're going to do this. So can you give me some examples of some setbacks, and how did Alnylam respond to these kinds of setbacks?
Well, the simple fact that it's 16 years between founding the company and the first products that generated revenue, that then revenue has to grow before it's really significant. There was many different setbacks. The first was everybody was excited about RNAi. This was something that worked in the laboratory, could have worked to treat patients. But the attention span of the market from that perspective is about 5 years. So if you're not in sales within 5 years, then the market, the press starts to say, what's going on here? So that dynamic is always there. Then we had setbacks in an early clinical trial, which was begun. The patients were probably too far along, and the drug was, the RNAi design was probably not optimal. It wasn't optimal. We redesigned it. But we had a setback, and people were saying, the street was saying, this technology may not work. It may be complicated. You have to manage your costs. You have to be able to, and we did, downsize. You had to focus. We had to sharpen our focus on certain products to actually put the resources we had to those products. And we had to begin to finance in different ways. And one was to take some debt, and one was to sell some future royalties, and then others is to form relationships with companies that give up the rights to future income from products you're developing. But out of all of that, maintaining a focus, keeping your burn rate at a rate that doesn't take your future, but allows you to develop the technology, is management and science working together.
Because I do remember there was a phase 3 failure, and that was very shocking because at that point you were ready to start marketing, and so you had to rethink how to.
Well, it was pre-marketing, but it was, we saw a asymmetry in deaths in certain trial. Don't really know for sure why even today, but we withdrew the product from a clinical trial, stopped the clinical trial, and then ultimately redesigned it with a more active, more specific agent as we understood the technology better, and then ultimately got approval in that area. But, it's part of what
makes biotech so interesting.
Biotech such a complicated field, because when you go into patients, they're not lab mice. They live different ways. They do different things. They have different genetic backgrounds or life experiences. So you have to prove the technology is beneficial to patients, where they live and how their history, to really, be able to treat a disease.
Now, I want to riff on that. Clearly, these diseases are global. The same genetic mutations can occur in other populations. So international approvals and international markets are critical to companies like Alnylam, Japan, Europe, etc. So how did you go about figuring, how do I prove that this drug works in humans across the world, not just in the US, and then working with the regulatory agencies to get the approvals of these drugs, not just for the US, but also for international markets?
So when you begin your clinical trials, you do clinical trials in multiple countries simultaneously. Some in the US, some in Europe and other parts of the world. In hereditary TTR, there were a few founding populations, and in some countries, like Bulgaria, for example, there was a pretty significant population of patients with hereditary TTR. And that then became a country in which there were some clinical trials done. The same throughout different parts of Europe, Germany and others. And even in South America, there were trials, patients in the initial trials in that part of the world, because again, founder population. So it was international at the clinical trials, U.S. and Europe. The data was used to approach FDA for approval, and in their countries for their regulatory approval for treatment of patients. So to treat a rare disease like hereditary TTR, you need an international market and population to actually get the numbers that you need to do approval.
So from a statistical viewpoint, you need a large enough sample with enough control arm and treatment arm, and an international population gives you enough of that strength.
It gives you power. It gives you enough immediate power. And also it allows or provides the basis for that country's healthcare system to say, okay, there have been clinical trials done here and other parts of the world, and when we look at this altogether, there's clear benefit to patients, and we can approve the treatment. So you're international at the time you do your clinical trial, if you have the resources to do so, and it's very important to do it. And then you're international when you begin to market, because over half of the pharmaceutical market is outside the U.S. Ultimately, it's outside the U.S. But you need that market. You need to capture the income from that market to pay for this new technology that you're continuing to develop. RNAi is a platform, and it can be used and has been used for treatment of multiple disease. There are at least 7 or 8 different diseases that are being treated by RNAi now.
So that leads me to ask you 2 questions. One, you point out that RNAi is a platform, RNA interference is a platform, so many other companies are playing around in this space. So how do you deal with competition in this space? How do you decide to position yourself so that you don't either directly compete with them, or if you do compete, you're better than them? Can you shed some insights on how you went about that?
We were the first company organized to take RNAi, SI, small interfering RNA, to market to patients. The whole technology at 2002 was totally new. We had to convince the world that this technology works, all right? So we developed the technology, we published. Alnylam has published well over 200 papers over the last 20 years. Establishing technology works. It can be it works in humans. It works in models, animal models. And this is the clinical results that we will use to get approval. Different delivery modalities. Initial delivery modalities was the lipid nanoparticle. Then we developed, which has got to be administered IV. And that's an expensive way and a complex way to administer a drug. It's very difficult to do that in some locales. And we developed a sub-Q formulation.
Subcutaneous injection.
Subcutaneous injection, which you could do at home.
Right.
Just like taking insulin. That was much more convenient and had the surprising property that the way this RNA was developed to do that, it gave a very long half-life in patients. So we could potentially inject a drug or RNA once every 6 months, once every year. So it becomes then another platform that's very important for treatment of patients because it gives adherence to using the treatment. One of the big problems in pharmaceuticals is patients get pills and they take pills for a day or two, but or a month or two, or even a year or two, but if it's a chronic disease, you have to do it for the rest of your life if you're going to prevent that disease. And so adherence, by being able to deliver a drug once every 6 months, every time you go to the doctor, is really a very effective way to treat those diseases.
And my related question is, you began with rare diseases, but I suspect from what you've said earlier about pruning the portfolio, but also evolving the portfolio, that you have started emphasizing more prevalent diseases that are, larger populations, and so they have greater clinical and therapeutic benefit ultimately. So, how have you shifted in this sense, moving from rare diseases to diseases that are more prevalent in the population, for which RNA interference, small interfering RNA, can be used to treat disease?
Well, the objective, you have to keep the North Star.
Yeah, right.
The objective is to improve the lives of patients. And for example, hereditary TTR. Something like 40,000 patients worldwide are in that. But as we were developing TTR, it was clear that there was a chronic disease related to it that developed later in life, where the gene itself was not mutated, but something about the person and the conditions and whatever, that protein would begin to misfold and accumulate in the heart and cause heart failure. So here was a genetic disease that had a chronic disease complement, which a much larger population. In fact, it became clear as we were beginning to develop TTR that amyloid is a major cause of heart disease. So we then had a treatment focused on the gene that was important in this chronic disease. Now, we knew that if you didn't have the gene, there was actually very little consequence. So we could silence it over long term. In normal people, it did not have consequences. So we had a chronic disease, which was a much larger population than the genetic disease. And then you look at other situations, take Alzheimer's.
Mm-hmm.
We know that there's a protein from one gene that misfolds in the brain to cause Alzheimer's. So now you have, and there's some people who have a mutation in that gene that gets early onset Alzheimer's. Mm-hmm. But the broader population, that's a chronic disease. It develops over years as one gets older. Could one actually use RNAi to treat Alzheimer's? Because you know the gene, and we know in animal models that that gene can be silenced and there's very little consequence. So, there's another gene situation, chronic disease. You just have to then develop a delivery system to the brain.
To cross the blood-brain barrier.
Cross the blood-brain barrier, because your brain is protected from a lot of the elements in your blood because of a barrier that prevents it. So that's technology that Alnylam is developed. It's in clinical trials for early-stage Alzheimer's. So it's a delivery issue. It's trying to make an impact on patients and improving them. And I believe in the long term, this whole technology will be widely used in pharmaceuticals. Mm-hmm. It'll be another way of treating a wide spectrum of diseases.
Mm-hmm. Now, there's also CRISPR out there, CRISPR and similar companies that essentially modify genes, right? Yep. And that's a highly customized treatment from what I understand. You have to extract the patient's gene and then, figure out how to modify it and then push it back in the patient. And there's, immune responses. So sometimes you have to isolate the patient for 4, 6, 8 weeks in the hospital. And so the entire treatment might be individualized and highly expensive. Does silencing RNA as a path complement or make genetic modification using CRISPR and similar technologies superfluous?
Well, both CRISPR technology, as you just described it, and RNAi are gene-centric approaches to treating disease. Mm-hmm. Alnylam and RNAi silences that gene at the level of the RNA that is made from the DNA gene. Mm-hmm. And with CRISPR, you're modifying the DNA gene.
Right.
Now, you have 1 copy, 2 copies in every cell. And you have to introduce that CRISPR into the cell to then sort out that gene and then to induce a change in that gene that would make the gene functional, or introduce a whole new gene in the cell that has the non-functional gene. In both cases, you have to deliver a piece of DNA to the cell that has the disease function in tissue, in human tissue. And that's genetic engineering of somatic tissue, not germline. It's somatic tissue. You don't pass it on to your children. That technology will be very useful. But I think in many cases, it'll be easier to take RNAi and get a shot every 6 months or every year and deal with that, genetic problem. But we'll see. There is clearly need for this technology of CRISPR gene therapy technology. There are conditions in which it'll free patients from a life of just extraordinarily awful pain and other things. So it's going to benefit patients. Both of them are going to benefit patients. Both of them are the product of discoveries in really fundamental science, the most fundamental of science. And it's going to change healthcare to improve the lives of patients. And, if it's better with CRISPR, so be it CRISPR. If it's better with RNAi, so be it RNAi. It's a win-win.
Good. Now, Alnylam today has a market capitalization of around $60 billion. And currently it's around $2.5 billion in sales, perhaps a little bit larger. And it's got this initiative called P times 25, P5 times 25, which is become one of the top 5 biotech companies by 2025. So looking back, you've been working with this company for over 2 decades. And what are the key factors, if you want to summarize this, what are the key factors that have enabled Alnylam to become such a successful biotech company with very wide market acceptance, a $60 billion market cap, over $2.5 billion in sales, and a platform, RNAi as a platform that can continue to develop all kinds of very useful drugs for very difficult diseases?
You just said. You gave me the answer.
Okay.
It was a new platform that could treat many different diseases. We have 23,000 genes. Not all of them cause disease, but there are maybe 7,000 or 8,000 that will be identified as specific causing disease. This technology, we've developed 2 different ways to deliver it, one to liver, one to brain. Well, there's other tissues that, it's making progress on delivering, where different tissues such as muscle and other types of tissue in your body can be targeted to specifically take up these RNAis. In the brain, there is so much unmet need there, and all the way from Alzheimer's and Parkinson's to schizophrenia and depression and pain and just a whole host of things that we have very, I'd say, not ideal ways of dealing with. So that promise of, future markets, the fact that the company is going to turn positive in cash flow probably this year, the fact that it's got a robust pipeline of really talented people who are committed to making a difference in the world, and the fact that we've been transparent with the street, with scientists and others over that 20-year period, saying this is what we're going to do, by and large doing it, and even overproducing, I think has created a very promising foundation on which a company could continue to make remarkable advances.
So what advice would you have for young scientists, academics, professors in institutions, universities, as also biotech entrepreneurs who want to start a new biotech company, build on emerging biotech research, new research that's coming out, say membrane technology? Yeah. What advice would you have for them?
It's a complicated answer because things change. When Biogen was founded in 1978, it was the 3rd or 2nd biotech company organized. It was the whole industry didn't
exist then.
It exists.
Yeah.
And if you talk to someone from a major pharmaceutical industry, they would say, you don't have a chance because it's going to cost so much money to get to the point in which you're able to treat patients that you'll never be able to survive. And then, Wall Street started supporting this new technology even before it was going to be profitable, probably for 5 or 10 years. Venture capital stepped in and then found a promise to do it. So that was a different time. And then Alnylam was started 20 years later. The biotech history was there. Venture capital was there. Trained people were there. And a whole new platform and new science entered the field. At a time in 2002 was not a good market time. And it was only 2004, 2000, and later that the biotech market venture capital support post-establishment was there. Now it's a pretty sophisticated field. There are venture capitalists who have expertise, 40 years of experience, and there's people in hedge funds and finance who understand this. This is a whole category of investment. So young people and others who want to become involved need to learn how to meet people in the field, meet people in the investment, meet people who've had careers, identify venture type investments that will allow them to nucleate, and business people who can work with them as scientists to translate what they're excited about into something that can be useful to a pharmaceutical company or others that, will ultimately benefit patients. We're in a different world now. We have media. We have, machine learning. We have AI. Things are going to move more rapidly. Direct engagement of people is going to be more important. Healthcare is going to have to be re-engineered, the whole healthcare delivery system. And all of that will take innovation. It'll take very informed people working with physicians, with companies to make healthcare as efficient and effective as we can get it, because people need it.
Mm-hmm.
And it'll enrich their lives and their loved ones. And it's just an extraordinary thing to be involved in. You look back and you see people who have, very successful lives, and you've touched them in some way.
Mm-hmm.
And be able to do that, it makes life pretty special.
Phil, there's a new documentary that's going to be coming out about you. It's called Cracking the Code. I believe the director is Bill Haney, and it's going to be released on PBS this October. And it recounts your childhood, growing up on a farm in Kentucky, to now, becoming a Nobel Prize laureate, starting these two companies, Biogen and then Alnylam, and of course being a mentor to a couple of generations of postdocs and PhD students and other biotech companies where you've served on boards, for example. And also, of course, keeping a family front and center. So how have you balanced all of these different aspirations of yours, goals of yours, and had such an inspiring and successful career?
My career would not have been possible without the devoted support of my family, particularly my wife, Anne, and our 3 children. Personally, I program my time to make time, because it's important to me, for my family. and important for me to learn and work in my own personal research. I was department head at MIT. I was head of the Cancer Center. I founded a McGovern Brain Institute at MIT. I had a major focus at MIT. And then I had a major focus at Biogen, 25 years on the board, and Alnylam, 20 years on the board. So you have to balance your time. You have to have your goals clear to you and what you want, and you have to work. I mean, and it's just, it happened. I had a lot of people who supported me, and I depended on a lot of people to help me, and all throughout this. It really takes a lot of goodwill around people who work with you and you to them to be able to do these things. And yeah, I'm lucky. It's the only way I can describe it. I'm lucky.
Can you give me some more information about that documentary?
Well, the documentary is sort of three parts. It focuses on the early days of my career. I came out of Kentucky, small farm, as you mentioned, rural community. Went to a small college in Kentucky, and then to the University of Illinois, and then off to Caltech, and then Cold Spring Harbor before coming to MIT. And it follows that
trajectory,
and my wife and my life and all of that. And then it got a significant part that the biotech. How did biotech develop Biogen as an early company, and then Alnylam subsequently, and how that works with Wall Street and finance and all that. And then at the end of it, it highlights my academic Nobel research and what that meant, and the whole contribution of Alnylam's LNP to the COVID vaccine. So it's a very interesting documentary. I hope people view it and like it and take some from it in terms of why this is all important. The investments in research, basic research, the investments in biotech and its contribution to society, and the whole training and supporting people. It's a very important story from that perspective.
Yeah, I think that's very important, especially given some of the misinformation about what RNA is and what RNA can do. And I think this talk is very useful because it shows how RNA has the potential to cure really difficult diseases and improve quality of life for everyone.
It doesn't have the potential. It has already demonstrated it's real.
Exactly. Well put. This has been a wonderful, very enlightening conversation. Thank you so much for taking the time to come and talk to us, to our audience. You've been listening to International Business Today podcast. I'm Ravi Sarathy, and my guest has been Professor Phil Sharp, Nobel Laureate in Medicine 1993, founder of Biogen and Alnylam, and Institute Professor at MIT. If you've enjoyed this podcast, please do send us your comments and questions. I hope you'll subscribe to the podcast, and please do come back again for another episode of International Business Today. Thank you very much.